In this video, we will learn how to extract information from Seurat objects, which includes single-cell RNA-seq data or spatially resolved transcriptomics data. First, we will discuss why this is helpful in the analysis. Then we will get an overview of the structure of the Seurat object. Lastly, we will discuss how to use the tool in Chipster. Essentially, the object is an S4 object for single-cell genomic data or spatial transcriptomics data and its associated information. The data is stored in the object in these specific slots, which can contain the data in various formats, such as numeric matrices or data frames. To get a better view of the structure, you could imagine the object being like a drawer with different kinds of slots for different contents. Because of the complex structure of the object, accessing specific information inside can be tricky. There are various situations where the tool could come handy. For example, if you have some data and you're unsure if it contains all the parts you need, you can use this tool to get a general view of the object. Moreover, you can easily, for example, check whether SCT transform or the global scaling normalization was used to normalize the data. Next, we will take a look at the overall structure of the Seurat object. One of the main slots in the object is the assay slot. The assay slot includes the counts and the data matrix, as well as other slots for the highly variable features and for the standardized expression values for each gene across all genes. The different assays include the RNA, SCT, integrated, and spatial assays. Single-cell RNA-seq data always contains the RNA assay, where the raw counts are stored in the count slot. If the data has been normalized with the global scaling normalization, the normalized expression values are stored in the data slot of the RNA assay. The counts and the data slot are stored in two-dimensional matrices, where the genes run as the rows and cells as the columns of the matrix, as shown below in the graph. Furthermore, if the data has been normalized with the SCT transform, the normalized expression values are stored in the SCT assays, data and counts slots. The integrated assay you would have if you're doing multiple sample analyses and you have combined the samples using the integration method. Other slots in the object are, for example, the reductions, comments, and metadata slots. In the reduction slot, dimensional reduction information such as principal component analysis or others are stored. Each reduction contains the coordinates of the cells in low dimensional space, as well as the weights for the genes stored along the dimensions of the cell embeddings, as also shown in the row below. The comment slot contains all the arguments that have been run to the object. Furthermore, the metadata data frame contains additional information associated with the cells or the features of the data. Spatial transcriptomic data is stored in the object in a similar manner, but with some exceptions. The assay slots have the same structure, but instead of the raw counts being stored in the RNA assay, they are stored in the spatial assay. In spatial transcriptomics, it's also essential to know the spatial location in the tissue, and this is why the tissue image is also stored in the object in the image slot. Next, we will discuss how to run the tool in Chipster. As input for the tool, you can give the R object or an RDS file containing single-cell RNA-seq data or spatial transcriptomics data. And as parameters, you can choose which result tables to extract. As the result tables, you can get the slots text file, which gives an overall view of the information in the object. This file is automatically produced when running the tool. The other tables which you can choose in the parameters include a table of the genes and a table of the highly variable genes in the object according to the active assay. For example, if you have already run the SCT transform for the object, you will get a list of the filtered genes and not the ones in the raw data, which are stored in the RNA assay. Furthermore, you can choose to produce a table of the metadata data frame containing additional information associated with the cells or the genes in the object. These can include, for example, the subpopulation identity of a cell or gene variants. And in case you have spatial transcriptomics data, you can also extract the default tissue image stored in the object. Lastly, we will take a look how to use the tool in practice in Chipster. We can, for example, run the tool for this single-cell RNA-seq Seurat object that is SCT transformed by choosing the file and the extract information from Seurat object tool. You can find the tool both from the single-cell RNA-seq category as well as from the spatially resolved transcriptomics category. From the parameters, we can choose which output files to produce. We can choose all of them, but as we don't have spatial transcriptomics data, we cannot extract the spatial image. As the result, we get the following files. Firstly, we get the slots text file containing overall information of the object. You can, for example, see all the assays that are present in the object and check which of them is the active one. From the assays, you can also see how many genes and how many cells are present in the data. Moreover, you can check, for example, which dimensional reduction methods have been run to the object. Secondly, we get the tables of the genes in the object, now in the SCT assay, and the highly variable genes. Furthermore, there is the comments table where you can see the details of the SCT transform. Lastly, we also get the metadata table with information like the number of molecules detected within a cell and the number of genes detected in each cell.